Hi, my name is Ana Magos, and this is my answer to question number 1. The polymerase chain reaction, also known as PCR. PCR is used to copy DNA molecules via in vitro. This means that it's made outside the body or in a test tube. This is very useful for researchers because PCR replicates exponentially, so in about 30 cycles, billions of DNA are created. And let's keep in mind that the DNA molecules that are created are double-stranded. Epidemiologists used PCR back in 2014 to amplify the genomes of two Ebola virus outbreaks, and after comparing the nucleotide sequences, they determined that they were two different strains of viruses that caused Ebola outbreaks in Central and Western Africa. Another example in which PCR is used is in crime scenes. When DNA is found, it's taken to the lab to run PCR and replicate the DNA found in crime scene. Just in case that the found DNA is messed up, investigators have a backup. Now, I'm going to talk about the 3 steps that are required to form PCR. Step number 1 is inactivation. Double-stranded DNA has to be heated at 94 degrees Celsius to break the hydrogen bonds between the base pairs. We can see it has opened up right here. Now, I'm going to talk about step number 2, priming. DNA primers are needed to bind to DNA sequence. DNA polymerase allows the DNA replication, and the DNA nucleotides A, T's, G's, and C's are added to target DNA. We can see the DNA primer, the nucleotides of the DNA polymerase being added to the DNA, and now the DNA has to be cooled down to 65 degrees. This allows priming to occur and form a double-stranded DNA molecules. Now I'm going to talk about step number 3, which is extension. The temperature is raised to 72 degrees to increase the DNA polymerase replication rate. We can see that the two strands of DNA are being created. And then to keep forming PCR, all the steps have to be repeated: inactivation, priming, and extension. PCR can be done by using a thermocycler, which is an automated device that performs PCR. In order to use a thermocycler, a special DNA polymerase known as Taq DNA polymerase must be used. Taq polymerase is derived from a bacteria, Thermus aquaticus, and is used because it's not denatured at 94 degrees Celsius. So the thermocycler does not need to be replenished with DNA polymerase continuously. Thank you.